Good morning, dear students. Today, myself SHEKHAR, we are going to discuss about the module number 4 of chapter 11, Biotechnology, Principle and Processes. Here we are going to discuss about the steps involved in recombination DNA technologies. The steps are: first, isolation of desired DNA, means to separate out the desired DNA. We get the fragments of this desired DNA by restriction endonucleases. Isolation of these desired DNA fragments. Ligation means to tie, to attach of DNA fragments into the vector. Transferring the recombinant DNA into the host. Culturing the host cells and extraction of desired products. Here within the diagram you have to see that we are going to attach the desired DNA with plasmid body, which is called recombinant plasmid. Now, how the extraction of DNA or isolation of desired gene will take place? The cells of organisms that contain desired genes are collected. The DNA of these cells is extracted by using refrigerated centrifugal technology. The content of the cells is treated with enzymes like chitinase, lysozyme, cellulase, ribulase and proteases, because it contains a large number of impurities in the form of RNA, lipids, etc. Finally, chilled ethanol is added to purified DNA. The isolation of gene is done by shoot gun method. In this specific restriction enzyme is used to cut and isolate desired gene. The isolated gene contains two sticky ends. The desired gene is isolated by using gel electrophoresis technique. Gel electrophoresis, as the name indicates. Gel, we are going to use certain medium kept into electric field, then by the phoresis process it is going to be separated. The steps which are going to be used in gel electrophoresis: first, the fragments obtained after cutting with restriction endonuclease are separated by using gel electrophoresis. Electric field is applied to the electrophoresis matrix. Commonly we use aerosol gel. The negatively charged DNA fragments move toward the anode, means positively charged anode. Fragments are separated according to their size due to sieving properties of aerosol gels. Smaller the fragments move faster than longer one. Then we stain these fragments with ethidium bromide, followed by exposure to UV radiation are used to visualize the DNA fragment. So when we kept these stained fragments in ultraviolet radiation, then only we becomes able to see these fragments. DNA fragments are visible as bright orange colored bands in the aerosol matrix. These bands are cut from the aerosol and extracted from the gel piece. That process is called elution. DNA fragments are purified and used in construction of recombinant DNA. Here, this one is the device in which we are going to carry the process of gel electrophoresis. Thank you. Have a nice day.